A major challenge in biology today is mapping genotype to phenotype. A key aspect of the mapping function is gene regulation, and it is increasingly evident that we need to capture the dynamics of gene expression if we are to understand the complexity of its regulation. This is particularly challenging in multicellular organisms, in which spatial as well as temporal aspects of gene expression need to be monitored. The growing root of Arabidopsis is particularly well suited for real-time imaging and analysis of gene expression in both time and space, but the challenge lies in conducting live microscopy on a large number of specimens grown under controlled conditions. Our goal was to obtain time series recordings of gene expression at tissue-specific resolution in a large number of plants with minimal human intervention.
We needed to develop a device in which many seedlings can grow in parallel, and roots can be directly imaged using confocal microscopy without manipulating individual plants.
Towards this end, we developed the RootArray, a device that allows for automated, high-throughput, live confocal imaging of Arabidopsis roots. The RootArray is a microfluidics device that is built using additive manufacturing technology or stereolithography. This device is more than a scaffold for imaging plants. It functions as a growth chamber in which the plants germinate and grow, and where they can be exposed to various treatments prior to and during imaging. Its core component is a translucent and biologically inert phytopolymer scaffold that contains up to 128 wells for individual seedlings.
This is a standard growth chamber, just like you have in your research lab. You can use this space to grow seedlings to adult stage plants. Or you can use this space to house your RootArray. Not so much space is needed to house your RootArray. The platform's quite small. This chamber can grow 64 seedlings at once. We also have a module in which you can grow 128. Here you can see we have 12 experiments running simultaneously.
Each of the RootArray's wells is filled with agar in which a single seed is planted. Ports at the side of the RootArray supply liquid and gaseous medium into the root and shoot chambers, respectively. Upon assembly, the complete RootArray is sterilized with a bleach solution and connected to a peristaltic pump, which supplies the gas and liquid to the system. This setup enables a continuous exchange of growth media and the application of chemical compounds, stains, and other exogenous treatments.
Each RootArray can be reused multiple times. We generally use one device for 10 to 20 experiments. Assembled RootArray are mounted vertically on racks under continuous light, so seeds can germinate and seedlings can grow.
After germination in the RootArray, roots grow through the agar into liquid media where they are imaged using a confocal microscope. Software automatically detects the individual roots, follows them as they grow, and takes a continuous series of images of each root over time. Thus, roots are grown, treated, and imaged in the same environment. This allows for observation of dynamic changes in gene expression and cellular morphology in response to a stimulus. An automated image analysis pipeline straightens the roots, aligns the images to an atlas of root cell types, and quantifies fluorescent intensity as a measure of gene expression. The final result is a quantification of cell type-specific gene expression over time for individual roots grown together in controlled conditions. The RootArray enables high-throughput observation of dynamic changes in gene expression and protein localization in individual Arabidopsis roots at a level of spatial and temporal resolution that cannot be achieved through traditional methods.
We expect that the RootArray technology holds promise for a broad spectrum of applications.
For additional details about this system, please see our recent publication in Nature Methods or visit the websites of the Benfey Lab or Grassroots Biotechnology.